Hello Miss Kina, we are from Group 3. Our presentation is discussing about high fructose effect in cancer cells. This is the outline of the presentation. First, we are going to talk about the introduction. Unexpected fructose metabolism in cancer cells have been observed. Thus, the researchers conduct a research to observe the fructose effect in leukemic cells, and they found several facts. The aim of the research was to understand the mechanism of fructose metabolism through SSP on leukemic cells and find a promising approach for acute myeloid leukemia. Next, we are going to talk about the results.
The first experiment is to investigate the metabolism and synthesis of multiple strains in high fructose conditions. It is done by adding salt into two 13 carbon isotope fructose and glucose medium and using NMR for isotope tracing. The result is that THP1 and Kasumi1 has similar fructose consumption as glucose consumption, and the increased production of serine and glycine in fructose while decreased production of lactate in fructose.
The next step was to identify what factors drove the fructose metabolism. In this case, the expression level of GOOD5 was observed. Based on the results, it was found that MYM13 and K562 cells had lower level of GOOD5 than THP1 and Kasumi1 cells. Figure C and D showed cells with low level of GOOD5 produce higher ratio of glycine to lactate in fructose media in comparison with glucose media. In conclusion, GOOD5 expression level determined the level of fructose consumption, which also regulated the serine synthesis at reflux.
In this experiment, they want to check whether SSP metabolism program exists in normal hematopoietic cells by investigating the GLUT5 abundance and fructose metabolism. They found that CD34 plus and MOLM13 cells had similar amount of GLUT5 and that their glucose fructose metabolism were similar to GLUT5 high cells. But they did not show higher flux of SSP and fructose rich condition. Based on that, SSP may be a potential therapeutic target.
Fructose is mostly metabolized by ketohexokinase rather than hexokinase in small intestine and liver. The aim of this experiment was to identify the fructose metabolism pathway in leukemic cells. This was done by inactivating ketohexokinase in MYM13 and THP1 cells, and later on hexokinase 2 was inactivated in acute myeloid leukemic cells. Based on the results, it was confirmed that fructose is mainly metabolized by hexokinase 2, since inactivation of hexokinase 2 led to a significant decrease in lactate yield in both MYM13 and THP1 cells.
The next experiment was to prove the hypothesis that GLUT5 low leukemic cells are more dependent on SSP. The inhibition of PHGDH by NCT503 and CBL5884 led to low glycine level, which hampered the cell growth. However, as on figure D, the CD34 plus cells amount remained the same on control and treatment groups. Furthermore, NCT503 caused apoptosis, which was detected by annexin V protein, as seen on figure E. The activation of pyruvate kinase also hampered the cell growth by reducing the SSP flux. Then they investigated the cause of higher SSP flux. They compared several enzymes downstream expression level in fructose and glucose-rich mediums. However, the differences could only be found on the ratio of NAD plus to NADH. Lower NADH level was observed in fructose-rich condition, which explained the high oxygen consumption rate because NADH is oxidized to NAD plus for PHGDH enzymatic reaction. The addition of lactate to the medium as expected decreased the ratio and low SSP flux could be observed. Meanwhile, the addition of pyruvate increased the ratio and higher SSP flux could be observed.
In this experiment, they want to identify metabolic benefit from high SSP flux by investigating three most important SSP functions in cellular metabolism. They found that antioxidant supplement did not alter glycine level in fructose. A lesser amount of glycine was effective in TMAKG supplementation, and that TMAKG supplementation will not affect cell growth. They also found that glutamine contribute higher to AKG pool and downstream TCA metabolites in fructose-rich condition. Based on that, they believe that SSP had an essential role to support TCA anaplerosis in fructose-rich condition.
In this experiment, the researcher aimed to test whether high fructose concentration does affect the dependency of leukemic cells on SSP in vivo. Experiment given a mice with leukemic cells, MLM13 in high fructose environment, and an observation of MLM13 metabolites and SSP in vivo. This experiment showed that the engraftment were more effective and leukemic cell would be more dependent on SSP in the fructose-rich environment.
And now we shall move into the discussions. In tumor microenvironment, glucose levels are low as the nutrients are low, so leukemic cells needs fructose to survive. Based on the research, leukemic cells use SSP in high fructose conditions. The switch to SSP is caused by increase in NAD plus and NADH concentrations. The restriction of SSP through the inhibition of PHGDH, the coenzyme, is crucial in controlling the cancer.
Conclusion: SSP has an important role in leukemic cells by utilizing fructose as fuel in glucose deficiency. In dependent condition to SSP, PHGDH enzyme is a great target for acute myeloid leukemia treatment. And here are some recommendations for the further study. That is all from us. If you have any question, please contact us. And here's our reference. Thank you for listening.